6.6. So, good evening, everyone. This is Kanika Gupta, and I'll be starting the session by 6:10. Let everyone join, and then we'll start with the session. Is that okay with you all? Admit Kathy. Okay, everyone. So, good evening, everyone. I'll start with my brief introduction to you guys. So, I am Kanika Gupta, and I have done my bachelor's and master's in zoology from Punjab University, Chandigarh. And right now, I'm pursuing my PhD from IIT Delhi in interdisciplinary science. So, we'll keep this session in a way that I'll give you a brief recap of what you guys must have studied through the videos in the last 1 week, and then we'll take your doubts and obviously the assignment that you must have solved. If you guys have any doubt in that, and we can take that also, and any other doubt if you guys have. Okay, so I'll be starting with that recap of what you guys have studied in the last 1 week. Are my slides visible to you guys? Just give me a yes in the comment section, or you can raise a hand also so that I can know that you guys. Okay, perfect. Thank you. So, first of all, as you guys have seen in the videos, also, the sir must have taught you that we'll first know what a homeostasis means. Homeostasis, in a very general way, we call it as a balance. The balance that is maintained in our body regarding each and everything, either it is sugar level, iron level, or any kind of balance in terms of blood pressure, in terms of the amount of blood, the amount of water in the blood, everything is balanced in body, and it should be maintained in that way only. Otherwise there can be many problems in the body if that balance is disturbed. So that is what you guys must have studied, that the average blood sugar level for a healthy adult without diabetes, this is, we are talking about an adult body, a normal adult, it should be between 70 to 100 milligrams per 100 ml. If it is disturbed somehow, for example, like if it exceeds by 100 milligrams to 140 milligrams, then that is a condition we call it as diabetes. Okay, and if it drops somehow below 70, then that is also a very problematic condition. Okay, so this is what you guys must have studied in the videos. Next thing is, the perfect definition for homeostasis would be, it is a mechanism that maintains a stable internal environment despite the changes present in the external environment. Because body has some mechanism, some cope up mechanism to adjust with these changes in the external environment. Basically, homeostasis is the balance in the internal environment of that particular person, on or we can say on in that particular body. The internal environment should be maintained. If there are some changes in the external environment, then body can adjust according to that, but obviously it should be maintained. Okay, so next thing, just a moment. Yes. So next thing, what we will study, obviously the term, the internal environment term was given by this gentleman. Sorry, the homeostasis term was given by this gentleman, Walter Cannon, and he was a professor at the Department of Physiology at Harvard Medical School. So, what he says about homeostasis is, the physiological system functions to maintain or regulate the condition of the internal environment within relatively narrow limits. If you see a chart, I'll show you in the next slide, that there are very narrow ranges for everything to be maintained in a perfect harmony, or we can say a perfect balance, which is thought to be the ideal condition for a body. Okay, there are very narrow ranges for that. And the homeostasis, the etymology for this is similar or standing still, that is staying the same. Whether there are changes in the external environment, the internal environment should remain the same. It should maintain its balance, its homeostasis. Okay, so this is what homeostasis means, and this the homeostasis is very important for a body to function. Why it is important? As you can see in slide, that there is a perfect, there is an amount which is said to be is important, or we would say is said to be a normal for any cell inside the body. For extracellular fluid, the sodium ion concentration should be maintained in between 142 milliequimolar per liter to the while in intracellular fluid it reduces to 10, and this is a balance which should be maintained. The external value shouldn't drop below 142, and the intracellular fluid value for potassium ion shouldn't exceed by 10. So this is what a balance which should be maintained, and this is the homeostasis which should be maintained inside the cell, or we can say in intracellular environment, so that the body can function, or we would say we'll start with the very basic entity, that is the cell. The cell can function in a proper manner, in a perfect manner. Okay, so these are some of the things which should be maintained in a way as they have been shown here. And this state in which the concentration of these different ions, or we would say glucose, amino acids, etc., will be in this concentration only, will be said as the homeostasis for that particular uh, body. Okay, so next thing is, as it says, most cells of an animal are exposed to internal environment, not the external environment. That is why the balance should be maintained in the internal environment, because external environment is not something which is in our control, but internal environment should be maintained for a body to function properly. Okay, so yes, homeostasis is a very important term and a very important phenomena inside a body. The major functional systems work together to maintain this homeostasis. For example, if we are talking about this oxygen, so there should be a level maintained of this oxygen inside the blood vessels. The blood which is flowing in the blood vessels should have a proper concentration for oxygen and carbon dioxide, in a way that when the exchange occurs at the cellular level, it can exchange to its maximum capacity. So cells can get maximum of oxygen while exchanging this oxygen and carbon dioxide, and it will help to perform the cell function properly. So yes, for example, जैसे यहाँ पे इन्होंने बोला है, lungs provide oxygen to extracellular fluid to replenish the oxygen used by the cells. Okay, and this extracellular fluids then further helps cells to uptake this, and obviously again the function to make it function properly. Another thing is about the blood sugar levels. Homeostasis says that the blood sugar level should be maintained around 90 mg per 100 ml of blood. It is said as the homeostasis for the body. Now, if it exceeds, like if you had any meal, then the level of the glucose instantly rises in your blood. Okay, it reaches up to 100, 120, 140. So in that situation, what happens is body gets a signal. Body comes to know that yes, the blood sugar levels are rising. It should be lessened down so that it doesn't become toxic to the body or the organs or whatever things it is coming in contact with, or whatever things it is they are using this glucose as. So what happens is, the when the blood glucose level rises after eating, after you take a meal, then the islets of Langerhanses, there's a group of cells inside the pancreas, in which the specifically the beta cells, come into action, and they release insulin. That insulin is actually working in a way that it will make different cells in the body to uptake that glucose, that raised level of glucose in the blood. It will make those cells to uptake that glucose, so that the blood glucose level can be maintained. Okay, so in liver it functions in a way that when the liver cells uptake this glucose is converted into glycogen in the liver cells, and that glycogen is actually the storage form of glucose inside the liver. And whenever there is a requirement of glucose inside the body, like when the blood glucose level drops below 70 mg per 100 ml, then again a phenomena works in which the alpha cells of these islets of Langerhanses, they'll come into action and they'll secrete glucagon. That glucagon will make the glycogen release the glycogen, it will, you know f- it will release the glucose, and that glucose is then further used by body for different functions it need the glucose for. Basically it is needed for providing the energy to the body to perform different functions. So yes, this glucagon will then perform the function of release of glucose from the storage forms, and then that will again maintain the homeostasis of the blood sugar level. Okay, so this is how this this different hormones, or we will say different organs, are involved in maintaining the homeostasis inside the body regarding the sugar levels in the blood. Okay, another thing is it is the control of temperature. Sometimes we will see that the temperature of the environment has risen up. So in that scenario, when the temperature outside, like in the external environment, is rising up, it will somehow affect the internal temperature, or would say internal environment also. Then also there are different organs or different mechanisms running inside the body which will come into action and will help in maintaining the temperature inside the body. It should be maintained at 37 degrees Celsius. Outside temperature can rise up to 48, 50, which generally happens in summers. But the internal temperature of a body should be maintained at 37 degrees Celsius, for which again there are control centers present in brain, which will signal different organs, or we would say different mechanism at different point of time according to the requirement of that situation, to maintain that temperature of the body. Like if it is too hot, it will make the blood vessels dilate, causing sweating, again making the temperature inside the body to lessen down to 37 degrees Celsius. And if it is dropping, the outside temperature is dropping below a certain temperature, like if it is cold outside, then the blood vessels will constrict, and instead of sweating, will there will be a shivering in body, which will cause the generation of heat inside the body, and that heat will again help to maintain the internal body temperature for that particular person. Okay, so yes, there are control at every step inside the body to maintain a proper homeostasis, to let the different hormones, different proteins, different macromolecules present inside the body to function properly and keep the normal functioning, the vital functions going on. Okay, so next thing, again, there's a set point. There should be some mechanism involved which tells the body that yes, this should be maintained this way only. There is a set point at each and everything inside the body which tells that yes, it should be like this only. This can't change. For example, the temperature, it should be maintained at 37 degrees Celsius. It shouldn't drop below 37 degrees Celsius. And if it rises by above 37 degrees Celsius, then again it is a problematic condition which we generally call as fever. And if it rises too high, then it can cause death also in some situations. So yes, there is a set point for everything inside the body. This set point is somehow connected to the control center. When the set point is disturbed, there are different circuits present inside this control center. This control center is present inside the brain, in the central nervous system. So when there is some kind of disturbance in the set point of that particular person, in any way, then these circuits work in a harmony, and they will perform the sensory function also and the effector function. They'll send the signals in different direction depending upon what is the requirement at that situation. And these again, when this sensory function reaches this control center, it will send motor signals to effector organs or effector parts that the set point is being disturbed. There is some situation in which the things are not in a harmony, the things are not maintained in a way that it should be maintained, and again the necessary actions are taken inside the body by different organs or different mechanisms involved. Okay, so set point is a very important thing inside the body. For example, like uh, we know that there is a particular blood pressure inside the blood vessels when blood is flowing. That blood pressure should be maintained the way it is, but sometimes the blood pressure is also disturbed. Then how come the brain knows that yes, there is a disturbance inside blood vessels which is causing this change in the blood pressure inside the blood vessels? So this happens like the vagus nerve is the one which is involved also along with the glossopharyngeal nerve. So what these things do is like the vagus nerve has just mm, yes. So as I told you that this vagus nerve has its nerve endings inside this aorta walls. So when the nerve endings are actually what? These are baroreceptors. Baroreceptors are what? These are the receptors which can sense the change in pressure. Okay, so these nerve endings are actually what? Baroreceptors. So when they observe some kind of a change in pressure, they send that signal to the medulla oblongata, or we can say CNS. In CNS, as I told you, there are control centers. When they find like there is some change in the blood pressure inside the aorta, then it sends the signal. Sorry, it sends the signal further that the pressure is either declining or it is too high to inside the blood vessel. And according to that signal, whether it is too high or too low, different kind of hormones or different mechanisms come into action inside the body, which will then try to maintain the blood pressure back to normal. If it is not some kind of abnormality, if it is like some situation, for example, there is some fight or flight situation. Do you understand what is fight or flight situation? Okay, I'll explain. It is a situation like you are in a situation and some dog is running behind you, or you are exercising. Let's take it that way. If you are exercising, it is not a fight or flight situation, but yes, when you are exercising, then your body is doing an activity which is strenuous, and it is not something you do you keep on doing whole day. Okay, so in that situation the blood pressure increases. It increases in a way because some parts of the body require more oxygen to perform that strenuous activity. In that situation, there are these baroreceptors will send a signal to the medulla oblongata, or we'll say the CNS, that yes, the blood pressure is rising, which is not a normal condition for a body to function. So it will try to maintain that blood pressure back to normal, so that the different parts of the body can get sufficient oxygen also, and the blood pressure is also not too high, so that it won't damage the blood capillaries, because blood capillaries are very thin walled structures. Very high blood pressure can in some situation can cause damage to those blood capillaries, which is also not a very good condition for body. Again, when there is a fight or flight situation, as I told you earlier, in that situation also body will try to keep that homeostasis, so that there's no damage inside the body. There is no I would say much change in the internal environment of the body which can cause a larger damage to the body. As I told you, different organs work in a harmony to maintain that internal environment, so that the internal environment remains the same as it was, uh, whatever the external environment is. Yes, the internal environment, the set points, I would say, are a little bit flexible in a way that they can shift a bit according to the external environment. It is not like they are very rigid. If it is a situation in which a set point should be changed, like in night, in the day, there are different mechanisms working in a body. It obviously it shifts, but there is a set point for every situation inside the body. Okay. So, next thing is the changes in the physiology that are responsible to changes in the external environment. As I told you earlier, also, that okay, so as I told you earlier also, that there are changes in the physiological conditions depending upon the external environment, but body will, as I told you earlier also, when the temperature rises. Okay, this is the situation of a temperature. Body will send a signal that there is an imbalance, and according to that imbalance, there will be response from the uh, different organs. Okay, so this was what we understood in the homeostasis. And the next thing that we understood is the muscle physiology part. But before going into that, I would like to ask you guys if you guys understood till now, if you guys could recall something that you guys had studied in the lectures, or you want me to slow down my pace, or you want it to be fast enough. You can tell me if there is any problem. uh, if there is any problem, just write in comments, and if you want it to be continued, then I'll continue. Anyone, if any question till now? Okay, if there is no question, then I'll move forward with the muscle physiology. So yes, this is a very interesting topic, muscle physiology, as this is something which actually we use in daily life, but we don't come to know that there are many things involved in that one movement. If you are moving a single finger of yours, then there are many things involved in moving that single finger. Okay, so before going into the details of the contraction and relaxation and everything, first we should understand the structure of a muscle. So yes, muscle is actually I would say, is a bundle of different muscle cell, not different, I would say, sorry, many muscle cells that are enclosed together by an outer covering. That outer covering will then for that outer covering is actually what, a connective tissue. This connective tissue is actually running all along. It will connect this muscle to the bone. But the one thing that you should keep in mind here is that in muscle fibers, as I told you, that this white portion is what, connective tissue, this red portion is your muscle cells. Okay, so muscle cells are running all along, but at a certain point of time the muscle cells stop, that they don't go beyond that. What is actually connecting your muscle to a bone is this white connective tissue which we call as tendon. Okay, so just remember this thing, that a connection between a bone and a muscle is through a tendon, not like a muscle is directly connecting to the bone. Okay, this is sometimes gets confusing. So this is what, the tendon is what connecting bone to muscle. Muscles is not directly getting connected to a bone. Okay, and as I told you, it is a bundle of many muscle cells that are coming together to form this bundle. Okay, single muscle fiber cells may be as long as 30 centimeter, or the diameter is generally 10 to 100 microns in diameter. Okay, and next thing is this. So as I told you, that this is the single muscle cell, and these many muscle cells together come together to make the whole muscle. Okay, and this muscle cell, if you can see, this is also a skeletal muscle. There are three different types of muscles, you guys must have known that. So this is a skeletal muscle, and if you can see, in this single muscle cell there are many nuclei. So what are these many nuclei? These are actually many muscle cells make a syncytium. Syncytium means is that the plasma membrane in between these different cells is dissolved, or we can say vanishes, and this whole different cells together make a single cell together, in which the nuclei remain same. The content inside the cell is still there, but the plasma membrane that was separating this one cell from the another cell has dissolved, or it has merged with each other, making these different cells a single whole cell, a muscle cell. Okay. And this muscle cell also has a satellite cell attached to it. Satellite cells are actually, these are the stem cells. These cells come into action when there are some kind of injury, or there are some kind of damage to these skeletal cells. Then this satellite cell, as I told you, these are stem cells. Stem cells are actually undifferentiated cells. So these satellite cells will differentiate according to the requirement, that it will differentiate either into the muscle cell or whatever the requirement is, as there are only muscle cells involved. So it will differentiate into a new muscle cell to take the place of the damaged muscle cell. So satellite cells are actually the stem cells inside the this whole arrangement. Okay. Now, when we are talking about a single muscle cell, now just keep this thing in mind if you guys are following me along with me. So, it is a bundle of different muscle cells, and when we are talking about a single muscle cell, then it is a bundle of many myofibrils. Okay. So what happens is, there's a single myofibril, there are many myofibrils. These many myofibrils come together to form a single muscle cell, and the many muscle cells come together to form a muscle, a proper muscle. Okay. So just keep this thing in mind, don't get confused. Myofibril is a different term and muscle cell is a different term, but muscle cell or muscle fiber are one and the same thing. Okay. Muscle fiber and muscle cell are same, but myofibril is different from muscle fiber or muscle cell. Okay. Many myofibrils come together to form a single muscle cell. Okay. So this is a structure of a muscle fiber. What happens in a muscle fiber, or I would say a muscle cell? As I told you, many myofibrils. These are all myofibrils. So these many myofibrils come together to make this muscle cell. The outer covering is called as the sarcolemma, and the fluid present inside, that is called as the sarcoplasm. In a cell, generally we call it cytoplasm. So here we are talking about muscles. So this is sarcoplasm. Okay. Okay. चलिए, sarcoplasm. So, as you can see, there are many striations present in these myofibrils. These striations are actually what we will see further in the presentation. That these are actually what different parts of the structure of a sarcolemma. What is sarcolemma? I'll tell you. Sorry, sarcomere. I'll tell you further in the presentation. So these different striations. These are actually light and dark bands. Dark bands are called as the A band, and the light bands are called as the I band. Okay. If you can see the structure, yes. So if you can see the structure, these are actually the dark bands. The myosin containing area is the dark bands. Dark bands because these are thick, and these are called as the dark bands because when they were actually discovered, then when the light was being passed through them perpendicularly, they were coming out to be dark. Also, the light when passed through it perpendicularly, it was coming out to be anisotropic. The refractive index was keep on changing. So these bands are these this dark bands are also called as the A band. Okay. Anisotropic band. And the lighter area or light bands are all also called as the I band or isotropic bands. Okay. Now in the center of the I band is present Z line. In the center of the A band is present the M line. And the area, as you can see in the structure, this is the red one is myosin, the green ones are actin. But if you can see in this dark band, there are this is the middle portion in which only the myosin is present, and no actin is present. So this portion is called as the H zone. Okay, this is called as the H zone. And the area between from one Z line to another Z line or Z disc is called as the sarcomere. This is a complete unit, and this when we'll see the function, you'll come to know that this whole will work as a unit, and this whole area will be involved in a contraction as a unit. Okay, in a when we'll study the skeletal muscle contraction. And if you can see, one more thing here is that these all actin and myosin fibers are arranged in a proper arrangement inside this myofibril. It's not like they are just laying haphazardly. No, they are arranged in a proper way at a proper distance from each other. This arrangement is very important for a skeletal muscle to function properly. Because of this arrangement, these uh, if you guys must have attended the lecture, so you must be knowing that these actually contract by a sliding filament theory. Okay, a sliding movement occurs between these fibers. So this arrangement, this geometrical arrangement is very important for that sliding to occur in a proper manner and these muscles to contract accordingly. Okay, so yes, this is what the structure of a myofibril is. Another thing is this myofibrils are actually uh, flanked by, or we would say are covered in sarcoplasmic reticulum. This sarcoplasmic reticulum is a rich source of calcium ions. Calcium ions are very important for a muscle to contract. So the calcium ions will, where from the muscle will get the calcium ion, it will be from this sarcoplasmic reticulum. Just a moment. Okay. Okay, जी. So this sarcoplasmic reticulum, and if you can see this tube-like structure here, just give me a moment. If you can see this tube-like structure here, these are actually T tubules. The function of these T tubules is actually to activate this sarcoplasmic reticulum, that yes, there is an action potential and the muscle must contract now. So these T tubules are actually the ones which will send signals inside the myofibrils that this is the time to contract. And obviously the mitochondria is, again, as you guys must be knowing, it is the powerhouse of the cell. So this myofibril also needs energy to contract. So where from it comes is the mitochondria. Okay, so the organization, as I told you earlier, this is the myosin filament, this is the actin filament, and if you can see this fiber, this protein running in the whole length of a sarcomere, this is actually the titin. What is the function of titin is titin actually maintains the structure of this sarcomere. It won't let it squeeze when the muscle is contracting, like it won't let it how would I explain to you guys, that it won't let it shatter. It will keep the things in place. It will maintain this structure of sarcomere, in short. Okay, another thing is, if you can see this protein running in purple, this is nebulin. Nebulin is actually running along the length of the actin. It maintains the position and length of the actin. Another protein is also involved in keeping the actin in position, that is this, sorry, I forgot the name. This is present tight, not titan. Sorry, I forgot the name. So yes, these are the proteins that are involved here. Apart from that, you guys already know. We'll go into the details of the myosin later. We'll see the structure so that you'll come to know that what are these small structures that are coming out of the myosin. Okay, so yes, first we'll see the structure of a actin. Actin is actually a polymer of G actin. What is G actin? G actin is just a moment. Yes, Anshit Poliye, you can turn on your mic. Anshul, do you have any question? So, okay. Okay. So yes, as I told you earlier, so actin is actually a polymer of G-actin. G-actin are globular actins, and when they'll come and arrange in a form of a fiber, then it is called as the F-actin. That is the fibrous actin or filamentous actin. Okay. So this actin also has another protein wrapped around itself. That is the tropomyosin. What this tropomyosin does is it blocks the myosin binding site on the actins, on the G actins, so that no unnecessary binding or disruptions occur inside this between these proteins. Okay. Another protein involved is troponin. What this troponin does is, we'll see in further slides. Next is myosin. Myosin is actually, it is a bundle of many myosin monomers coming together. Nearly 2 to 400 units of myosin monomers come together to form a single thick filament of myosin, which you can see in the center. Okay. So yes, it was the titin only, guys. Sorry, the protein I was, as I was talking about, the spring-like protein titin, which keeps all of the structure in position. Okay, and so this is what I've explained earlier. Also, myosin, the structure of myosin is this, that it has a tail. It has two light chains present in on the neck region of this myosin. These are actually the regulatory chains. Two pairs of light chains are there. So actually four chains. So these are actually the regulatory chains. It has sites. One is for ATP binding and other is for actin binding. You guys must have seen the structure, that why it needs the actin binding site. Okay, and we'll see it further in when we'll study the function. Okay, as I told you, the another protein that is involved is troponin on the actin. This troponin has actually three sites to bind. One, it binds to tropomyosin. Another site, it binds to actin, and on the third last site, it binds to the calcium ions. These calcium ions are actually important for a muscle to contract. How these calcium ions are important, we'll see further now. So, the mechanism, or we'll say the sliding filament theory, what it says, what is the mechanism of contraction of a muscle cell, a skeletal muscle cell, is this, that the first of all, there is the first thing that comes into action is that this myosin head is binding to the actin binding site on the uh, it's binding to actin through its actin binding site. Okay, on the globular actins in which the myosin binding site is present. Okay, and the ATP binding site is empty now. So this is a rigor state. That is, it is a stiff state in which the myosin is binding to actin, but it can't move because it doesn't have energy present. So what happens is, when an ATP molecule comes and binds through the ATP binding site on this myosin head, then it will come back to its original position. Now this ATP binding site has a protein present near this, which is a ATP, is an enzyme which will break this ATP into ADP plus inorganic phosphate, which will provide this energy to this myosin head, which will cause the myosin to bind on the actin through the actin binding site to actin. When it will bind to actin through this actin binding site, then after, what happens is, when inorganic phosphate leaves this, then the power stroke happens. In power stroke, what happens is, the myosin will make the actin slide over it, and it will try to move it inward. Okay, so this is the state which happens. Then after, when the ADP leaves this side, then the state of rigor happens, as I told you earlier also, that the state of rigor happens. And this is the state which happens when some death, when a person is dead. So what happens is, the energy content that was present already is all used, and then the muscles somehow stays in the state of the rigor, which is called as the state of rigor mortis, which then eventually settles down because the protein content of the body gets start getting degraded. Okay, so body again loosens up. Otherwise, the body is stiff because the muscles are in a state of rigor because it doesn't have any ATP attached to their ATP binding site. Okay, so in this way, this power stroke and this rigor happens inside the body, inside this sarcomere, and these actins will be moved inwards, like if it is myosin and if it is actin, so this myosin head will bind here on actin, and it will bring actin in this direction. The angle, if you can see that here it is a perpendicular angle when this myosin binds to actin, and when it slides it over through it, then the angle becomes acute, and the angles become more acute when ATP comes and bind here and the myosin releases the actin. Okay, so in this way this contraction occurs. Now how does the cell come to know that it is sarcomere comes to know that it is the time to contract? So it happens here is that the nerve endings present on the muscle fibers actually sends a signal in the form of this they'll get an action potential, which will then activate the acetylcholine granules present in the synaptic knob of these nerve endings, and these synaptic knobs, then there'll be change in the voltage, and the voltage gated ion channels will open, which will then further cause these acetylcholine granules to fuse with this synaptic membrane, presynaptic membrane, which will then release the acetylcholine. This acetylcholine will then further cause a change in the voltage gated ion channels. The sodium and calcium channels are involved here. Okay, so these voltage gated channels then further transfer this. This is actually the change of chemical signal to this electrical signal. ठीक है, तो what happens is this action potential further travels in the T tubules. In this T tubule is present the voltage gated calcium channels, which are also called as DHPRs, dihydropyrimidine receptors. Okay, so what happens is when these action potential reaches these receptors, then these are the voltage gated calcium channels, so they open. There's some change in the conformation of these channels, which leads to the changes in these ryanodine receptors, which are mechanically attached to these DHPRs, and then the change in the conformation of DHPR causes a change in the ryanodine receptors, which will then open the calcium channels of the sarcoplasmic reticulum, causing the calcium get released inside the sarcomeres. When this calcium is released inside the sarcomeres, this calcium goes and binds to the troponins. When the calcium binds to troponins, it will cause the tropomyosin to move aside from the myosin binding sites on the G actin. If you can see this, the tropomyosin is actually covering the myosin binding site on G actins. So when this calcium binds to these troponin, it will make the tropomyosin shift from this position, causing the site to get exposed, and the exposed site then gets binds to the myosin, and the muscle can contract. But this is very very fast it is occurring inside the cell, and there are also pumps present on the sarcoplasmic reticulum, which works in a uh, concentration manner. So it will then pump back the excess calcium present outside the sarcoplasmic reticulum back to sarcoplasmic reticulum lumen, so that the calcium can again get stored in the sarcoplasmic reticulum, and the muscle can again come back to its original position. So this all happens very fast. This all works in seconds or milliseconds way. Okay, so this is the whole cycle how it happens. That's an action potential in muscle membrane, depolarization of T tubules. As I told you, the action potential is traveling in T tubules that will open the sarcoplasmic reticulum calcium channels. The calcium channels will then release the ryanodine receptors, will then release the calcium inside the sarcoplasm, which will then further cause the calcium to bind to troponin C, and this troponin C will then cause the tropomyosin to move away from G actin, exposing the myosin binding site, and then this myosin binding site can get cross-linked with the with the myosin head, which has an actin binding site, and obviously the contraction can happen and reverse happens for the relaxation. Okay, so this is how it works, and this was it that I wanted you guys to recall. Now we can take your doubts. So anyone has any doubt, they can just tell me. Yes, Anshit, Ansh, boliye. What doubt do you have? You can unmute your mic and tell me.
मैम इसमें केमिकल प्रोसेस कैसे हो सकता है, जैसे मैं आप बताइए इलेक्ट्रिकल प्रोसेस
केमिकल
इसमें
प्रोसेस इज एक्चुअली एसिटाइल कोलिन जो है, मैं आपको दिखाती हूँ प्रेजेंटेशन जस्ट। देखिए, स्लाइड्स दिख रही हैं आपको?
जी मैम।
देखिए, व्हाट हैपेंस इज, यहाँ पे आप देख रहे हैं, दिस इज अ साइनेप्टिक नॉब। ठीक है,
ओके।
in this synaptic node we have this synaptic vesicles, and this in these synaptic vesicles, if you can see this mark, this is acetylcholine. Acetylcholine is a chemical.
केमिकल ओके।
Okay, so
न्यूरो
is
ट्रांसमीटर,
हाँ,
जस्ट लाइक ए न्यूरो ट्रांसमीटर।
yeah, it is a neurotransmitter, it's not like a
ओके
neurotransmitter, it is a neurotransmitter.
ओके।
So when there is an action potential, this action potential will cause these synaptic vesicles to get fused. This, we will call this membrane as presynaptic vesicle. Oh sorry, membrane. Why we will call this as presynaptic vesicle? Because this is synapse, this membrane is coming after synapse, so this is called as the postsynaptic membrane, and this is coming before, because from here the signal will pass to the synaptic area, and then it will move further in the postsynaptic membrane. So this is a presynaptic membrane. When these vesicles will fuse with this presynaptic membrane, then this acetylcholine, which is a chemical and a neurotransmitter, it will be released in the synaptic cleft. Okay,
ओके मैम ओके।
and this will cause these channels. There are ion channels present inside this postsynaptic membrane. So this will cause the action potential change here. This will cause the ion channels to open here, which will further send the signal through T tubules to sarcoplasmic reticulum in the form of an action potential. Now, if you can see here, there is no actual chemical traveling to these T tubules or sarcoplasmic reticulum, but there is a change. There is a change in the potential. There is a depolarization of the membrane, which is causing these receptors to further send the signal to sarcoplasmic reticulum that this is the time to release calcium, and this is the time for a muscle to contract. So this is how a chemical signal is getting converted into an electrical signal. Okay,
ओके मैम, थैंक यू।
you're welcome. Okay, anyone? If someone has any doubt left, they can ask me. Were you guys able to solve the assignment? Did you guys find any difficulty in solving the
If you
assignment?
don't mind, can you tell about how the acetylcholine is formation?
Sorry,
Formation of acetylcholine. Can you explain about it?
acetylcholine.
Hmm. Formation.
Acetylcholine is a
Formation.
neurotransmitter, which is a macromolecule, which is formed inside the body. That is a different mechanism. There are actually acetylcholine present inside this synaptic knob always in the synaptic vesicles. Okay, it is not like what happens here is it is the traveling of acetylcholine. Here it is not like acetylcholine is being formed here, then it is traveling in synaptic vesicles, and then it is further sending the signal. Acetylcholine is present here, which is formed inside the body, which is a neurotransmitter, which is again a chemical reaction that is happening. But what is happening here is this acetylcholine will travel in the synaptic cleft. This area is synaptic cleft, where where it will travel, and then it will send the signal further. Okay,
Okay.
do you get that, or do you want me to explain more?
Yeah, ma'am, I get it.
Okay,
Ma'am, what is a generation and conduction of a nerve impulse?
sorry.
What is a generation and conduction of nerve impulse?
Okay, so generation is it is actually when the nerve endings. Do you understand what are nerve endings?
Yeah, yes, ma'am. Nerve
Okay,
endings.
so there are many signals involved, as I told you already. That if you remember this diagram, I'll show you. So here, just don't see this set point thing, just see this thing, that there are sensors. What happens is when there are nerves involved, then it has a portion, a cell body, it has nerve endings, it has dendrimers, and it has an axon. So whenever a neuron gets a signal, it can be any form, like when we saw in this diagram, it was in the form of a pressure. These are baroreceptors. There is, if there is any change in the pressure, then these nerve endings will detect that change in pressure, and it will generate a signal. Okay, a nerve signal, it will be generated, which will travel through this axon is a part of a nerve cell. So it will travel through this axon in the form of an action potential. Okay, the change in ions concentration outside the
Okay.
membrane and inside the membrane, like inside the cell and outside the cell. So it will travel in the form of this action potential. It will reach the center where it is located. It will tell that yes, this problem has happened, yeah, this change has happened. Then like here the CNS, the brain is involved. Then there are many circuits are present inside the brain, which will then further send the signal back to the effector organ. Here the effector organ is heart. Okay. In the
Okay.
heart, they'll send back the signal that the pressure is either high or low. You should pump like more with pressure, or you should lessen the pressure. There are many things involved. So this is the generation of signal. When this nerve endings gets a signal, it can be in any form. Like for an example, when you touch a hot plate. Okay, so then heat is actually acting as an trigger for the nerve endings, which will further generate the signal and will pass through the axon to the cell body. Yes, that hand has touched the hot plate. Now the signal, when it reaches to CNS will respond it in a way that it will further send the signal, and the effector organ is again hand, because we have to remove the hand from the hot plate. It's not like the hot plate will remove itself. We have to remove the hand. Okay, so the CNS will send the signal back to our hand through a motor neuron, that the plate is hot, please remove your hand. Now what happens here is the signal has been sent to hand, but in hand, what is actually moving the hand is a muscle. So the signal is actually being sent to a muscle in the form of again action potentials, and as you can see, in the form of the acetylcholine, which will further cause the muscles to contract and will remove our hand. So in this way, a signal is generated and the signal is traversed. It all works through action potential. Changes in polarity, changes in ion concentration. Okay.
Okay. Thank you.
Any more questions? First, let's solve the assignment first. If anyone was having problem in solving the assignment, I'll just go through it quickly so that we can clear the confusion if anyone is having. So the first question was this. This was the structure given to you guys, and you were supposed to mark this. Okay. So the one, the thing which was marked as one, what we call it as? the glossopharyngeal nerve, which is also the ninth spinal nerve. Okay. Second is carotid sinus. Carotid is actually the carotid artery which sends the blood to the upper part of the body, basically to the brain. Okay. Third is your what? Third is vagus nerve. ठीक है? Vagus nerve is one of the longest nerve in the body. And fourth is what? Aortic
Aortic bioreceptors.
receptors. Do you understand the term baroreceptors? आपको पता है baroreceptors क्या होते हैं?
Just like receptors
Receptors पर किस चीज के लिए होते हैं
To receive the
baroreceptors?
signals, ma'am. To receive the signals.
No. Baro means change in pressure. Okay. These are actually receptors to sense the change in pressure. Baro means pressure. So that's why they are called as the baroreceptors. Okay. So these are actually the aortic baroreceptors. If anyone had any problem in solving this question, they can tell me. If anyone is having question in this question, then they can also ask me. Otherwise, I'll move forward.
No, ma'am.
Clear? Okay, then. Next question was this. So in this, I hope you guys were able to do this. We revised this thing in the today's class also. So one is the organ which is marked. One is what it is.
Pancreas, which intake
Pancreas.
the
Pancreas.
glucose.
Yes, yes, yes. And the two is
The cells intaking the glucose.
yes, right, right. Okay. Third is
The blood glucose level is 90 milligram per 100 mg of
per 100 ml
ml.
या फिर आप इसको per deciliter भी बोल सकते हैं, ठीक है? Okay. And the fourth is
The pancreas releases glycogen.
glucagon. Yes, glucagon. It is a hormone. See here, you should remember two things very clearly. That insulin is a hormone which will reduce the blood glucose level, and glucagon is a hormone which will increase the blood glucose level. Okay, these are antagonistic to each other. Okay, antagonistic means they are working in a opposite manner to each other. Okay, so this thing you guys should remember. Okay, so next question is this. What is the protein which is marked as 1? Anyone?
Triponin, ma'am.
No, it is actin.
Actin.
Okay, G actin, globular actin, which will polymerize to form the F actin या फिर fibrous actin, filamentous actin. Okay, so this is a G actin or actin we can say. Okay, okay. What is the protein which is marked as 2?
Troponin.
Right. Troponin.
Troponin, right? Troponin it is. It has how many sites?
3.
Three sites. One for
Troponin T, troponin L.
किस चीज से बाइंड करता है
I.
वो तीन साइट्स इफ यू कैन नेम देम एक साइट बाइंड टू ट्रोपोमाइसिन another site to bind to
Actin.
actin and third site to bind to
Bind to calcium ions.
calcium ions, right? And how many calcium ions can bind in that calcium binding site of
4.
troponin? Four, right? Very right. It is the four calcium ions that can bind here, and they work in a cooperative manner. If first calcium binds here, then it will increase the its site's affinity for other calcium to bind here. Okay, so it works in a cooperative manner. Okay, guys. So in this picture, if you can see, what is this thing which is marked as 1? Excuse me, guys. What is happening here? How the screen is getting changed? Hmm. So yes, again the thing is, what is this thing which is marked as one? What is this?
Greyhound.
Gray horn. This is a gray matter or gray horn of a spinal cord. Okay. Two is, what is this thing which is marked as two?
Um,
This is the dorsal root.
no.
Dorsal root of
Okay.
nerve. ठीक है? Because there are many nerves arising from this gray matter. So this is the dorsal root. Third is
Ganglion.
dorsal root ganglion, right? And the fourth is
Sensor.
sensory
Sensory,
neurons.
no?
Sensory neurons. Yes, it is a sensory neuron. Okay. चलिए और कोई क्वेश्चन किसी को पूछना हो लेक्चर में कोई चीज नहीं समझ में आई हो दे कैन आस्क मी कोई कंफ्यूजन है जो अभी तक मैंने प्रेजेंट आप लोग को रिवाइज कराया था उस पार्ट में कोई कंफ्यूजन है यू कैन आस्क मी एवरीवन okay now I'll ask you guys some questions I haven't written those questions but I'll ask you guys so what is the protein that is running all along the length of a sarcomere will answer this question what is the protein which is running all along the length of a sarcomere anyone
Nebulin, ma'am.
sorry
Nebulin.
nebulin are you sure
Yes, ma'am. It contains position length of actin. No, no, ma'am. Sorry. It
okay
is
so
for
once again
actin.
so which protein is that
Nebulin.
it is titin if you can see here this titin is running all along the length of a sarcomere okay this is a very
Okay,
large
ma'am.
protein which is involved in this nebulin is a protein which is running along the length of an actin एक्टिन प्रोटीन की लेंथ को जो मेंटेन करता है या फिर हम ये कहें कि जो एक्टिन के साथ प्रोटीन running along the length of actin molecule है that is nebulin but the protein which is running all along the length of this sarcomere is actually titin okay okay what is the function of z disc can anyone tell me what is the function of z disc
To hold the actin filament, ma'am.
to hold the actin filament very right another thing is
Stitch back after the sliding. The
one more we can say is the function of z line is to maintain the length of the sarcomere okay because the sarcomere length is defined as starting from one z line to another z line okay so z line actually demarcates the length of a sarcomere so it is somehow helping to maintain the length of the sarcomere also okay so this is the function of a sarcomere okay so if why the thick band is also called as the A band.
because it is an anisotropic band, ma'am.
Okay, and why it
Because
is anisotropic? How come they know that?
because the reflection is not the same in all directions, ma'am.
Yes,
That's why.
when the light is being passed through it perpendicularly, then the refractive index was keep on changing, causing a very anisotropic kind of pattern. So that's why this dark band is also called as the A band or anisotropic band. Okay. Okay, sarcoplasmic reticulum is a rich source of which ion?
Calcium.
Calcium ion.
Calcium ion. Very good, very good. calcium ion from sarcoplasmic reticulum is
DHP receptor and ranodine receptor.
ryanodine receptors. Very good, very good. So this is the ryanodine receptors and DHPR, as I already told you. They are attached to each other mechanically, and when there is a change in the conformation of DHPR, it causes the ryanodine receptors to also change their conformation, which opens the calcium channels from sarcoplasmic reticulum. Then this sarcoplasmic reticulum releases the calcium ion inside the sarcomeres, causing them to causing them to
Contract.
contract. Yes. So calcium ions are very important for a muscle to contract. Okay. Okay. What is the function of a satellite cell?
Which may renew the injured cell or damaged cell.
Okay. So what is the term given to these satellite cells?
Or stem cells.
Stem
Stem
cells.
cells.
Yes, these are stem cells which are very undifferentiated cells. So they can differentiate in whichever cell they want to. Okay. Okay, and any more questions you guys have? If anyone wants to
Can
ask.
you tell me about where the particular ATP can find, ma'am? Because in the lecture video, they tell that in the inch only the ATP will bind. But here the picture will be in the head. The binding site will be.
Actually, what happens is, if you can see it in the presentation, are my slides visible to you guys?
Yes, ma'am.
Yeah. So if you can see here, this is the structure of a myosin. Okay. And this is the head region, this is the neck region. Together we called it as hinge region also. So what happens is, there is this ATP binding site present in the head of myosin, where the ATP will actually bind. And here also the present the ATP is enzyme, which will help in converting this ATP to ADP plus inorganic phosphate. Okay.
Ma'am, but the source told that in that video, the source told that in the light chain only there is a binding site for ATP.
No, not possible. I have also, you should, I guess, you should listen the video again. There is some misunderstanding, because there is this ATP binding site, and sir also told about this ATP binding site, that in the myosin head, or we can say cross bridge, is present this ATP binding site, where the ATP actually binds, and it will further be uh, degraded into ADP plus inorganic phosphate, and then the released energy will be used in the contraction of the muscle. Okay, you listen the video once again,
Okay, got it.
and if still there is some problem, if still there is some confusion, we can take it up in the next lecture, in the next session of the doubt clearing session. Okay. Any more questions, everyone? I will try throwing you guys more questions so that you guys can practice. Okay. Everyone got the muscle contraction part clearly? If there is some confusion, they can tell me. I can explain the cycle again. The sliding filament theory, or the contraction mechanism of skeletal muscle. Any confusion, they can ask me. Okay, what is the difference between a myofibril and a muscle fiber?
Many myofibrils forms muscle cells.
Sorry, can you please repeat it again?
Many myofibrils form muscle cells
Yes,
together
myofibrils.
to form.
Yes. Myofibrils together come and arrange in a form of a bundle, which will further form the muscle cells. The outer covering of these myofibrils, the bundle of this myofibril is called as
Connective tissue. Connective tissue.
No, sarcolemma. Connective tissue
सार्कोलिमा
is the
मैम.
covering of that bundle of muscle cells, not the covering of these myofibrils. Okay, so this is sarcolemma, but the connective tissue that is present, that is covering the muscle cells, is what we call connective tissue. And if anyone can tell me what is the connection between a bone and a muscle is called as
Tendon, and through tendon there is a connection, right?
tendons. Yes, tendons it is called as. Okay, so always keep this thing in mind that muscles do not travel all the way and directly get connected to a bone. It gets connected to a bone through connective tissue, which is called as the tendons. Okay, so yes, if anyone can count and tell me how many actin filaments are present around a single myosin bundle. How many actins are present? If you can see the arrangement, it is actually six.
Around 6, ma'am.
Yes,
6.
six. So if you can see that this is a myosin filament, and surrounding it are the actin filaments, which are six in number. Okay, so they are arranged very geometrically. You won't find a difference of a 1 micrometer also in the arrangement of these actin and myosin filaments. They are so geometrically arranged so that the movement of muscle or the contraction of muscle can occur smoothly. If there is any disruption in this arrangement, then the whole functioning of skeletal muscle will be disrupted or affected. So this is all very geometrically arranged in a way that the myosin filaments can slide these actin filaments inside the uh, towards the M lines so that the muscle can contract, and when it is relaxed, then these actin filaments will move back to their position. Okay, everyone got this excitation contraction coupling clearly? Any confusion in this? If anyone has any question, they can ask me. Okay, so one question I have is, if you guys had listened to the lecture very keenly, then tell me that insulin acts on three type of cells, as sir has told. What are those cells?
Come again. Can
Sorry,
can you ask the question again, ma'am?
sir has told in the video also that when insulin decreases the blood sugar level, then it activates three type of cells
Beta cells.
for uptake of glucose. Beta cells are the cells which are releasing the insulin. I am asking that insulin will make three types of cell to uptake the glucose to reduce the blood sugar level. What are those three types of cells? As sir has told in his lecture. If you guys remember.
No मैम.
Sorry, can you again say that please? What are those three types of cells?
No, not remember मैम.
Okay, I'll tell you what are those three types of cells. These are the skeletal muscle cells, these are the liver cells, and third is the adipocytes. Adipocytes are the cells which store fats. Okay, so what the adipocytes will do, they'll uptake the glucose and store it in the form of the fats. In liver cells, it will be stored in the form of, in liver cells, the glucose will be stored in the form of
Glycogen.
Glucagon.
Glycogen.
Glycogen, sorry. It's glycogen. And in skeletal muscles also there is some amount of glucose which is stored in the form of glycogen only. Okay. So this is how the glucose uptake
What about in liquid cells, ma'am?
sorry in
What about in liquid cells? You told me
lymphoid
before, right?
cells.
Hmm.
In lymphoid cells the glucose is not stored. Lymphoid cells
Ma'am,
are
you told me the three cells which uptake
it
the
is
glucose.
adipocytes. The skeletal muscle cells, the liver cells, and adipocytes. Adipocytes are the cells which stores fats. Okay, and the glucose which is uptaken by these adipocytes, it will be stored in the adipocytes in the form of fats. They'll convert it into fats. Okay.
Okay.
Okay, Ji. The next question that I'll ask you guys is, mm, next time I'll prepare some quick kind of thing for you people. What can we ask? Still, I'm asking you guys. If you guys have any doubt, then you can ask me. And apart from that, okay, what is the function of a T tubule? Tell me what is the function of
After
a T
the
tubule?
depolarization occur, the signals is passage to the DHP receptor and they'll open the retinine receptor too for the
Okay, so
calcium.
if we want to know the function of T tubule in a single line, what we will say?
The passage of stimuli.
Yes, it is. The function of T tubules is to pass the signal inside the myofibrils for the contraction of these myofibrils, for the contraction of sarcomeres. So T tubules are actually the ones which will send the signal inside that it is the time for contraction of the muscles. Okay,
Ma'am, I have one doubt, ma'am.
yes, please ask.
Can you tell me about the retinine receptor? Sir told in the lecture video about retinine in particular compound. It is a poisonous alkaloid, that and all. I didn't understand that part. Can you explain it briefly?
So in you are asking me about the rhinodine receptors, right?
Yes, ma'am.
So there is this alkaloid which is present in which we get from a plant. The that is the alkaloid is named as the rhinodine. Okay. So what happens is when this rhinodine binds to these rhinodine receptors, so it will block these receptors. So they won't be able to function properly, and if it won't be able to function properly, then the calcium won't be released from the sarcoplasmic reticulum, right? And if it won't be released from the sarcoplasmic reticulum, then the contraction won't happen.
Paralysis?
Paralysis possible, quite possible. Okay, one more
Okay.
thing I want to add in here is that do you guys remember that the neuromuscular junction, and we have the acetylcholine receptors in the postsynaptic membrane which detects
Yes.
the acetylcholine and then open the voltage gated channels. So the acetylcholine receptors here are nicotinic acetylcholine receptors. Why they are nicotinic acetylcholine receptors? Because these receptors can also bind to nicotine. Okay, nicotine is a
Okay.
compound which we get from what? From which plant? Tobacco plant. Okay, so if these receptors has an affinity for that nicotine, and if there is this nicotine binds to these receptors, then it won't go away from these receptors very easily, causing these receptors again to get blocked with this acetylcholine, although the acetylcholine binding sites are different in these receptors than the nicotine receptors, but still these receptors has an affinity for nicotine. So these are called as the nicotinic acetylcholine receptors. Another form of these acetylcholine receptor is muscarinic acetylcholine receptors, which has the affinity for muscarine. ठीक है?
Must.
This is a different form of acetylcholine receptor. There are two forms, that is nicotinic and muscarinic. Okay.
Okay.
These are voltage gated ion channels, the nicotinic acetylcholine receptors. The muscarinic acetylcholine receptors are actually G protein coupled receptors. That is something we'll further study in the detail in course. But you should know that there are two types of acetylcholine receptors. Okay.
Nicotine is also G coupled, ma'am.
No, it's voltage gated. As you can see here, these are nicotinic and these are voltage gated. Got the point?
Yeah.
Okay. So, what is the power stroke? If anyone can tell me.
Uh,
What is a power stroke?
Ma'am, when ATP binds the myosin, it start to bind the actin molecule to move towards the H band. After that, it relieve. That's the power stroke.
No. The power, this is all the binding of the myosin to actin. That is right. But power stroke is when this inorganic phosphate, when this ATP is binding to ATP binding site, then there is ATP is present there. This ATP is
No, ma'am,
released.
it's converted into ADP and inorganic
Yes,
phosphate.
yes, inorganic phosphate. Now, when this inorganic phosphate is released from here, at the same time, this myosin head starts the movement, like it will try to pull the actin inside towards the H band, and when this pulling is happening, that situation is called as the power stroke. Okay.
Mhm.
After that, when this ADP is also released, then the position stays the same, because this myosin head does not has the energy to get released from this actin. Okay, so it will
No, ma'am.
stay the same way, and it is called as the Riger state.
Uh,
And when again this ATP comes and bind to the ATP binding site, then this myosin head will be released from the actin, and it will come back to its original position, and it will be prepared again for another cycle of contraction. Okay, so power stroke is actually when the actin is being moved inside by the myosin head, the slided inside by the myosin head. Okay, this is what the power
Ma'am,
stroke.
please repeat the power stroke.
Sorry,
Please repeat this.
repeat kar do. Okay, so what happens is, we'll start from the one. Okay, to know the cycle, what happens actually. What happens is, when this myosin, you guys already know that myosin head has an ATP binding site. Okay, so when this ATP comes and bind to this ATP binding site on the myosin head, till then the myosin is detached from actin. It is not attached to actin. Now, in this ATP binding site, near the site, there is a presence of an enzyme which is an ATPase. Okay, so ATP, what will ATPase do? ATPase will break this ATP into ADP plus inorganic phosphate. When this ADP and inorganic phosphate is formed, then the energy is released, which is taken up by this myosin head, and it will bind to this G actin, or we will say actin filament, in the myosin binding site. यहाँ तक समझ आया सबको?
Yes, ma'am.
ठीक है. Now, when this ADP and inorganic phosphate is still there in the ATP binding site, and the myosin head is also connected to the binded to the actin filament in the myosin binding site. Now what happens is the inorganic phosphate is released from this ATP binding site. ADP is still there, but inorganic phosphate is released. At the same time, the myosin head will bend and it will bring the actin along with it in a sliding way. Like this is myosin and this is actin. And when there is this head hanging, when this head is binding to this actin and energy is given to this, then it will bring this actin along with it. Myosin is still in its place. It will bring the actin along with it with the help of his head, which is bound to the actin. So when this myosin head will bring the actin, it will move the actin, slide the actin. That situation is called as the power stroke. Okay. And then after power stroke, what happens is ADP is also released from the ATP binding site. Now, this myosin head is still bound to the actin, but it doesn't have the energy to get released from this actin binding site. Okay. So it will stay in that position only. This position is called as, this state is called as Riger state. Okay. Then again, when as the site is still empty, the ATP binding site is still empty. So when a new molecule of ATP comes and binds to this ATP binding site, then this myosin head is released from actin and it will come back to its original position and will be prepared for the next cycle of contraction. Do you got my point now? Do you understand
Yes,
what
ma'am.
power stroke is? Okay, perfect. If you can understand what a power stroke is. Okay, what is the structure? I would say, what is the thing involved in bringing the calcium levels of sarcoplasmic reticulum lumen, bringing the levels of calcium back to normal. The normal means the lumen of the sarcoplasmic reticulum has a very high calcium concentration. So, what is the thing that is involved in bringing the levels back to the normal? If anyone can answer the question. Can anyone tell me what is
Can you repeat the question, ma'am?
my question is, when the sarcoplasmic reticulum has released the calciums on activation, so what is the thing that is involved in bringing the levels of sarcoplasmic reticulum back to high for calcium or normal for calcium in sarcoplasmic reticulum lumen? What is the thing?
Are you asking about the bringing back of the calcium from the muscle cells, ma'am?
Yes, yes, yes.
Yeah, by the help of the calcium pump, ma'am.
Yes, they have these calcium pumps present in the walls of sarcoplasmic reticulum, which will work in a concentration dependent manner. And when they see that there's a difference in calcium concentration uh, between the lumen and the inside of this in the sarcoplasm, then it will pump back those calciums to sarcoplasmic reticulum lumen so that they can be used again. Okay.
But it also needs ATP, right, ma'am?
Yes, it also needs energy. It can't do it without energy. It requires energy to do that. Okay. And I hope you guys must have understood the events that occurs in the excitation contraction skeletal muscle in the skeletal muscle. It works in a very regulated manner in a way that one step will occur after the other, otherwise the second another step won't be able to happen. Okay, the action potential will travel through the T-tubules. The T-tubules will then catch up that action potential and will activate the DHPRs. The DHPRs will undergo a change in the conformation. The change in conformation in the DHPRs will be sensed by the ryanodine receptors. Then the ryanodine receptors will undergo the change in conformation, which will open the calcium channels. Calcium channels will release the calciums inside present inside the sarcoplasmic reticulum lumen to the sarcoplasm, or we will say to inside the sarcomere, where they will reach and bind to troponin. When they'll bind to troponin, troponin will cause the tropomyosin to shift from its position in a way that the myosin binding site will be seen, will be I would say exposed, and this myosin binding site, then when it is exposed, then the myosin will bind through its head, which has an actin binding site to this actin, and then the contraction will happen. Okay, so this all works in a very regulated manner and in a step by step events. It is not like anything can happen anytime. It works by very step by step regulated manner. Okay, and you guys got the concept of a set point. I hope you guys must have got that. Apart from that, this muscle contraction thing is a very interesting thing, and it is very important to understand this skeletal muscle contraction, so that you can understand further the cardiac muscle contraction and all. So if there is still any confusion, you can ask me, but please don't keep any confusion in mind for later, because it gets confusing. Can read further, it gets confusing sometimes. So ask me away if anything you don't understand, and if there is something amiss, you can ask me now. You guys do understand what a syncytium means, and why these muscle cells are multinucleated? Can anyone tell me what a syncytium means, and what is the meaning of these multinucleated? Do you guys understand that? Okay, if you guys understand that, that's very good. But A band, I band, you guys understand that the optical properties are not same in all directions, and the isotropic property or I band having the same refractive index in all directions in a muscle cells. Okay, that's why they are named so. A band, I band. And you guys must know a sarcomere starts with a Z line and end with a Z line. In the center of a band is present the M line. And what is an H zone? If anyone can tell me what is an H zone? What is an H zone in a sarcomere? Can anyone tell me, please?
Where the only myosin protein are present.
Yes, the H zone is present in the A band only, but this region is the region in which only myosin filaments are present, no actin filaments are there. Okay. Okay, so you guys understand the H zone
Ma'am,
very
can you
well.
tell me about the full form of H zone, ma'am?
Sorry,
Why they call that H zone?
sorry to tell you, but I also don't know why they are called H zone. They are just named so.
Okay.
We don't know why they are called as H zone. They are named in that way only. Okay.
Yeah.
Anything else you want to know? Sarcoplasmic reticulum, you understand the sarcoplasmic reticulum is present around these myofibrils. Okay. sarcoplasmic reticulum are actually the T tubules are actually passing through these sarcoplasmic reticulums so that they can get attached to the ryanodine receptors and then can get an access for the calcium release. If you can see the structures. Also, the T tubules are running from one end transversely, from one end of a muscle cell to the other end of the muscle cell, but in a transverse manner. So the action potential is traveling all along the T tubule. Okay, that is how the action potential is traveled. Proteins, you must have understood the nebulin, the titin, myosin, actin. Okay, these are the four important proteins. There are other proteins also which are involved, but these are the main four proteins which are involved and which are generally studied. But yes, obviously many other proteins are also involved. You guys must know this titin is traveling all along the sarcomere, and it is important to maintain the structure of a sarcomere, or we can say the length of the sarcomere. Nebulin is running all along the actin, not the whole sarcomere. It is actin only to keep in check the length of the actin and also to keep the actin in position. Okay, so actin and uh, myosin are the proteins which are actually involved in contraction directly, but nebulin and titin are the ones which are helping them and also help in maintaining the structure of this sarcomere. Okay, so you should remember these proteins correctly. Okay, one more thing that I forgot to ask you guys. What is this protein present in the Z disc or Z line? It's a very important protein. If anyone can tell me, what is the protein that is present in the Z disc or which forms the Z disc or Z line? What is the protein? If you guys have heard the lectures, then sir has talked about this protein.
Ma'am, Desmond?
Yes,
Ma'am, Desmin, ma'am.
yes, it's desmin. Desmin is also very important protein, which is the protein which helps the actin to bind at one end, like at Z line, because it is the desmin only through which the actin is binding. Okay, so desmin is also a very important protein involved in maintaining the structure of a sarcomere. So you should keep in check the desmin. And here there are two pair of chains, but in total four chains, four light chains are involved. Okay, these are regulatory proteins. These are regulatory in function. These light chains are regulatory in function. You should keep in mind that there are two pairs, but in total four chains. Okay, and this is called as the tail of myosin. These are the heavy chains. Actually, there are two pairs of light chain and one pair of heavy chain. So in total, how many chains of proteins are there? How many chains of proteins?
Three. Three.
Six, ma'am.
Six. Three pairs, but in total six chains of proteins, two heavy chains and four light chains. These two heavy chains are demarcated in a way that it is called as the tail region, it is the neck region, and it is the head region, or together we can call this neck and head region as the cross bridge also. Okay, we can call it as a cross bridge also, but this is the head region of the myosin heavy chain. This is the tail region, this is the head region. So you always, when someone asks you a question, read the question carefully. If they are asking about the pair, the total number, and all, always keep these things in mind. These are very small, but very important things when you are solving a question. And one more thing is, if you can see that there is this arrangement of myosins, if you can see that they are coming together to form a bundle, but the myosin heads are opposite to each other. If you can see that the arrangement is this, these are in different direction, these are in opposite direction, and also in arrangement also they are opposite to one another, and they work independently, as sir must have told you in the lecture. So yes, class, if anyone have any question, they can ask me. If still there are some questions that you want to ask, you can ask in the next lecture also. But if anyone wants to ask now, they can ask me. Just keep in mind the function of different proteins involved and also the location of different proteins, because it is very important when solving a question. Also, there are blood supplies present in the muscle cells. Otherwise muscle cells को nutrients कैसे मिलेंगे, है ना? So blood supply is also there in the muscle cells. Okay, one more thing is, the bundle of muscle cells which arrange together to form a whole muscle. So this single bundle is called as the fascicle, which which is covered by an outer layer of connective tissue called as the perimysium. Do you get the point? This is perimysium, and a single muscle cell is covered by endomysium. When these many muscle cells come together to form the bundle, which is called as the fascicle, it is covered by an outer layer of connective tissue called as the perimysium. And are these visible? And the outermost covering, which then further makes the tendon also, it is called as the epimysium. You should know these three layers: endomysium, perimysium, and epimysium. Okay, you should know which layer is covering which part of the muscle.
Ma'am, what about vesicles?
Vesicles, as I told you, it is a bundle of muscle cells. Okay, and the covering, the connective tissue covering that is covering this vesicle is perimysium. देखो, are you able to see? This is
Sure.
the fascicle, and this layer that is covering is perimysium, wrapped by perimysium. Okay, and the ones which is covering the muscle fiber are endomysium, and the one which is covering the whole muscle, it is and अपनी ये epimysium, sorry. And this now, as I told you, the muscle cell won't go all the way to bone to get attached, but it will halt at some point. But the connective tissue moves further and it will attach the muscle to the bone. So the connective tissue here involved and the covering which is involved is epimysium, which will make the tendon. Okay. Any other confusion or any other question if anyone wants to ask, they can. This muscle anatomy, this physiology and anatomy, you should understand very well. If you won't get the arrangement, then you won't be able to understand the functioning also. Read this thing and understand this thing very clearly. If still there is any confusion, you can ask me. If still you don't understand anything, there is some confusion in the anatomy part, you can ask me. Because the arrangement of a muscle is in a way, it is very geometrically arranged, very precisely managed, so that it won't hinder the functioning of other fibers. Okay, if one fiber is missing or displaced from its place, it will obviously hinder the other fibers working, and the contraction will be affected, or functioning of the muscle will be affected. So it is very well geometrically managed or arranged. Acha, the single cell which is involved, which further makes syncytium to make a whole muscle fiber or muscle cell. So the initially the single cell which makes the syncytium is called as what?
Myoblast.
Myoblasts. Yes, these are the myoblast cells. So you should remember these terms. Don't get confused in between these terms. Myoblast is the cell which was initially present, which further came together with other myoblasts and made the syncytium, and that syncytium then made what? A muscle fiber or muscle cell. Okay, and these myoblasts are mesodermal cells. You should keep in mind these are mesodermal cells, and this muscle fiber or muscle cell, it is actually a bundle of what? What are the tiny fibers running all along a muscle fiber is? What it is?
Myofibril.
Myofibrils. Yes, it is myofibril, which forms the bundle to make a complete muscle cell, and these muscle cells then form a bundle to make a fascicle. Then many fascicles come together to make a whole muscle. Okay, and the covering of a fascicle is called as? The connective tissue is called?
Endomysium.
Sorry, what do we call it as? It's
Endomysium.
no, it's perimysium. I'm asking about fascicle. I'm not asking about a single
Perimysium.
cell. It's perimysium. When I'm asking about a muscle fiber or a muscle cell, then it is endomysium. And when I'm asking about the whole muscle, that is the different bundles of fascicle will get arranged into a muscle. Then the covering is epimysium. Okay. So yes, class, any more questions, any more confusions, you can ask me. Otherwise, we can stop this meeting now. You guys can leave the meeting if still there are no questions involved. If you don't have any other questions, then you can leave the meeting. We'll stop this meeting now. Thank you so much for coming, and we'll meet next week then.
Thank you, ma'am.
Thank you.
Thank you, ma'am.